In this lesson, we are going to label carbohydrates as either D or L enantiomers. We're going to draw the mirror image of a carbohydrate molecule. We're going to learn to distinguish between mono, di, and polysaccharides, and how polysaccharides function as complex carbohydrates. And we're also going to recognize how carbohydrates determine blood type. When we look at the name of carbohydrates, we will often see the designation of D or L. This tells us which mirror image of the molecule we are working with. To identify something as D or L, we look at the last chiral carbon, which are highlighted here in yellow, and look at the position of the OH group. If the OH group is on the right, we designate that as the D sugar. If it's on the left, it's the L sugar. It is the D sugars which are most often found in nature. The pattern of whether the OHs are on one side or the other determines the identity of that particular carbohydrate. For example, in glucose, we look at the 4 chiral carbons in the middle of the compound. Notice that on the first chiral carbon, the third chiral carbon, and the fourth chiral carbon, the OH groups are all on the same side, and on the second chiral carbon, it's on the opposite side. The same is true for both versions of the structure, but this pattern is what determines something as a specific carbohydrate. Again, you are not responsible for memorizing the structures of any particular carbohydrates, but you should be able to designate them as D or L. When we look at monosaccharides, we're going to look at the name and see the prefix mono, which means one, and saccharide means sugar or carbohydrate molecule. So in a monosaccharide, we will have one carbohydrate forming that compound. Note that there are different ways to draw carbohydrate structures. The one we will see most often is the one on the right using the linear Fisher projection. However, in the body, carbohydrates actually fluctuate between a chain compound and a ring structure. And 2 examples of the way we can draw those ring structures are shown on the left. You will not need to convert between the chain and the ring structures. However, you should recognize that the structures on the left also represent a carbohydrate molecule. Because we only see the one ring, we know that this is a monosaccharide. When we look at disaccharides and polysaccharides, we see structures that are a little bit more complex. So di means 2, so 2 sugars or 2 rings in our structure. So disaccharides have 2 rings in their structure. Poly means many, so we see many of these rings in each of our polysaccharide structures. If you look on the lower right side of the screen, you'll see brackets with an N. This N tells us this structure is going to continue on for N units. So this is a way to represent a very large molecule without having to draw the entire thing when each carbohydrate is exactly the same in a repeating pattern. Nutritional carbohydrates can be divided into simple and complex carbs. Simple carbs are things like sugar, which is found in many foods, and starchy vegetables, such as potatoes. These simple carbs are broken down very easily in the body and often give us that sugar rush after we eat a large amount of them. Complex carbs have more nutritional value, they are slower to digest, we maintain that fuller feeling longer, and it takes longer for our body to break them down. These complex carbohydrates have up to thousands of monosaccharide units in one molecule. Carbohydrates also allow us to determine blood types. Red blood cells have units of carbohydrates attached to them. When we look at type O, for example, we see the pattern of the maroon to beige and a green. That same pattern is seen in every other blood type. Although every other blood type has additional carbohydrate units, it still has the same pattern that is in O. As a result, O is known as the universal donor. When O is given to patients of another blood type, it doesn't see that as a foreign substance because it matches up with at least part of the carbohydrate units attached to the other type blood cells. When we look at type AB, we see it has the most complex pattern of carbohydrate molecules. However, when we look at the pattern in that, we can find segments that match O, A, and B type blood. As a result, AB is the universal acceptor. The carbohydrates tell us about the O, A, B, and AB types. The plus and minus are determined by proteins that are on the surface of the red blood cells.